Okay, welcome everyone. Now I am going to discuss type 2 hypersensitivity reaction. So I hope you are clear with type 1 hypersensitivity reaction. I have discussed type 1 hypersensitivity reaction in detail. Fine. What is type 2? It is also called antibody mediated hypersensitivity reaction. Whenever some antigens enters the body, it will interact. I am using that word again. It is just an interaction. The antigen-antibody interaction is responsible for triggering inflammation. This will interfere with the normal functions. I am telling again, no tissue injury. No tissue injury, but inflammation present. No tissue injury, but inflammation present. Okay. There are a lot of diseases which are associated with type 2 hypersensitivity reaction. Lot of diseases. Fine. Now, this type 2 hypersensitivity reaction may occur in three different ways. Number one, opsonization and phagocytosis. Number two, complement and FC receptor mediated inflammation. Number three, antibody mediated cellular dysfunction. Okay, these are the three different mechanisms by which this reaction occurs. First is opsonization and phagocytosis. You know what happens whenever there is an antigen antibody interaction. Finally, the antigen will be destroyed by phagocytosis. I have told you in chapter inflammation, for making these antigens more tasty, I am using the word more tasty, yummy. Okay, yes, yummy or tasty. These cells will be opsonized by some IgG antibodies or C3b and C4b complements of the complement pathway. As a result of this opsonization, these antigens will be made more yummy or tasty. Hence, phagocytosis can easily happen. Remember, 2 points. IgG antibodies, C3b, C4b complements are responsive. They are the classical opsonins. Finally, phagocytosis of the opsonized cell occurs. Fine? Yes. Or if there is complement activation, what will happen? You know MAC. I have told you membrane attack complex. This membrane attack complex will how it is going to destroy the antigen? It will drill. Okay, it will drill the membranes, the lipid bilayer. It causes osmotic lysis of the cell. Fine? Yes. Also, this may occur in another way, that is called antibody mediated cellular cytotoxicity, ADCC. Okay, ADCC, that is antibody mediated cellular cytotoxicity. Fine. And via the NK cells and macrophages. Okay, am I right? Yes. And remember, in such a case, there won't be any phagocytosis. Here comes the role of what? Here comes the role of NK cells. You know, NK cells, they will, I'm writing here, you will learn in microbiology, NK cells will destroy cells via perforins and granzymes. So I hope this is clear. Perforins and granzymes. So these are the two enzymes which will cause some perforation or drilling in the membrane, and finally it causes cell death. So what are the two? So far I have discussed two pathways. Number one, opsonization and phagocytosis via C3b receptor. Opsonization, phagocytosis. Finally, phagocytosis will happen. This is one mechanism. Second mechanism, the FC receptor may cause complement activation, C5a, C3a. Finally, this will induce neutrophil enzymes and reactive oxygen species intermediate. Here comes the very, very important question. Which of the following types of or mechanism of type 2 hypersensitivity reaction is mediated via ROS, reactive oxygen species, ROS? The answer is complement and the FC receptor mediated inflammation. Very important. Okay, we will see in lot of examples. Okay, we will see a lot of conditions for each one of this. Fine, don't worry. So this is the second meaning. So in the second mechanism, via the activation of some chemotactic agents called as C5a, which will further causes migration of monocytes, increase vascular permeability. They are responsible for what? Tissue damage. What they will produce lysosomal enzymes, okay, and finally causes tissue damage. This is the second mechanism. Each mechanism we have some examples, we will see. The third mechanism is nothing else but what we call it as what? Immune mediated cellular dysfunction or antibody mediated cellular dysfunction. Here for an antigen, some antibodies will be produced. In antibodies will be directly produced against the antigen. Remember, in this mechanism, there is no cell injury or inflammation. Remember, these antibodies which are produced to the antigen, against the antigen, they may either increase the production, or they may even decrease or inhibit the production. We have two classical examples of antibody mediated cellular dysfunction type 2 hypersensitivity reaction. Number one is myasthenia gravis, where the autoantibodies are produced against the acetylcholine receptor. Acetylcholine is inhibited. We have another one condition, as you can see in this picture, that is the antibodies are produced against the TSH receptor. And remember, this TSH receptor is going to further aggravate. It will cause more release of T4, T3. That is what we call it as Graves' disease. So two conditions, either it may inhibit or either it may promote. So these are the three important mechanisms. By which type 2 hypersensitivity reaction happen. Fine. Okay. Let me say some examples. Okay. Let me say some examples. One second. I will discuss this table separately. Because you should know in detail about all these examples. Okay, one second. I will discuss this image separately. Okay, one second. Yes. We are going to discuss this image separately. Fine. I told we have three mechanisms: opsonization, number 2, complement mediated, number 3, antibody mediated. Okay. Autoimmune hemolytic anemia, autoimmune thrombocytopenia, autoimmune hemolytic anemia, and autoimmune thrombocytopenic purpura. These are the two conditions where we will get opsonization and phagocytosis mechanism. Here what is happening? Opsonization and phagocytosis of red cells happen. Okay, the patient will present in autoimmune hemolytic anemia. Here the antigens, that is here what is happening? Phagocytosis of red cell is happening. Here antibody is produced against what? Red cell membrane antigen, which may be either a Rh blood group antigen or an I antigen. You will see in hematology. Fine. Clinical, how the patient will present with hemolysis and anemia. Whereas in autoimmune thrombocytopenic purpura, AITP, here the antibodies are produced against platelet membrane proteins. They are glycoprotein 2B and 3A. Glycoprotein 2B, 3A. Each one is an MCQ. I'm going to write here VVIP table. Fine. Okay. This will also cause opsonization and phagocytosis of platelets. The patient will have bleeding manifestations. So two examples. I have used red color. Okay, for this. Red color for this. Once this is done, let us see the examples of complement mediated. We have only one example mainly, that is good pasture syndrome. I am writing with green color. In good pasture syndrome, the antibodies are the complements, I can't use the word antibodies. The complements are mainly targeted against some non-collagenous protein in the basement membrane of the kidney. In the basement membrane of the kidney, as well as in lung alveoli. Lung alveoli. The patient will have nephritis, lung hemorrhage. This is the one and only example. One and only example. Then, let us see other examples. What is the antibody mediated? We have 3 examples. I am using violet color. Number 1 is Pemphigus vulgaris. Pemphigus vulgaris. In Pemphigus vulgaris, the antibody is produced against the what? Proteins of the intercellular junctions. In the skin epidermis, we have desmosomes. We have desmosomes. Here, antibodies are produced against some proteins called as desmogleins and desmocolins. Desmogleins and desmocolins, which will cause intercellular adhesions interruption. The patient will have skin vesicles or bulla, rupture of the skin. Then, second condition is myasthenia gravis, where you already know antibodies are produced against acetylcholine receptor, which will down modulate the receptor, inhibit acetylcholine binding, causing muscle weakness and paralysis. Already known thing. Next, Graves' disease, third one, TSH receptor will be stimulated. Stimulation of TSH receptor. TSH is stimulated, hyperthyroidism, increased T3, T4. Increased T3, T4 will be there. Fine? Yes. So these 3, we have so far discussed the examples of 3 important mechanisms. There are some other examples which we can fit into miscellaneous groups. Miscellaneous group. That means they won't suit accurately to any of these 3 mechanisms. But still they are examples of type 2 hypersensitivity. Number 1 is Pernicious anemia. I am going to draw with red color. Pernicious anemia, antibodies produced against intrinsic factor, Castle's intrinsic factor of the gastric parietal cells. There will be decreased absorption of vitamin B12. The patient will present with anemia and abnormal erythropoiesis. Number 2, acute rheumatic fever. Acute rheumatic fever. Acute rheumatic fever. In acute rheumatic fever, what? The antibodies will be produced against the streptococcal wall antigen. Remember, the antigen of the streptococcus mimics the antigen of our own myocardium. As a result, what will happen when the body attacks the streptococcal antigen? The body will harm its own myocardium, resulting in myocarditis and arthritis. This is what we call as acute rheumatic fever or rheumatic heart disease. Fine, you will learn much more in microbiology. Then third one is nothing else but what we call as vasculitis caused by ANGA, anti-neutrophilic cytoplasmic antibodies. ANGA, anti-neutrophil cytoplasmic antibodies. Okay, here the neutrophil granule proteins, antibodies are produced against the neutrophil granule proteins. As a result, neutrophil degranulation will happen. That means the neutrophils will be abnormally activated. This is going to cause vasculitis. So I am going to write it here. Number 1, this color is nothing else but opsonization. Okay, number 1 and number 2. Then green color, number 1 and in green color we have only one number. In green color we have complement. Then in this color, that is 1 and 2 and 3, we have antibody mediated. And in red color we have 1 and 2 and 3. We have miscellaneous mechanisms. Fine. Okay, I hope this is very clear for you. This table is VVIP table. No doubt it's the VVIP table. Please open your eyes when you approach this question. Please open your eyes. Fine? Okay. I hope this is very clear for you. This is about type 2 hypersensitivity reaction. This is about type 2 or antibody mediated hypersensitivity reaction. I told you that type 2 diabetes mellitus, it is an example of type 3 hypersensitivity reaction, whereas type 1 diabetes is an example of type 4 hypersensitivity. This is one of the very frequently asked and repeated PGMCQ question. Thank you.